Okay. Assalamu alaikum, everyone. My name is Rida Hussain. I am a second-year medical student, and I will be teaching you guys about brush border digestion and absorption. It's a mouthful. Let's move. This is the content we will be covering. So I will be telling you an introduction, a background about the brush border in itself, different sites of absorption and methods of absorption. Then we're going to be talking about specifically how carbs are absorbed, proteins are absorbed, and lipids. And after each one of them, we're going to be talking about a complication regarding the absorption of them. We move. Okay, so what is the brush border? The brush border, basically, it is the part of the small intestine, which is where most of absorption takes place. This is where 95% of water is absorbed and a lot of the nutrients are absorbed. So, because most absorption is taking place over here, the brush border has to be um, fit for his job. So the advantages of a brush border would be like there is more interaction of the enzymes, so more digestion occurs over here. There is faster absorption. This is because the surface area is increased due to the microvilli being present. Um, it is also, lastly, it is also sterile. So what this means is that the distance between the microvilli, as you guys can see here, can you guys see my pointer? Um, wait, I want to point. Okay, over here. Can you guys see my pointer? Yeah, thank you so much. So the distance between the microvilli over here is smaller than the dimensions of a bacteria. So because it is smaller than the dimensions of a bacteria, it really doesn't allow for the bacteria to be absorbed into the enterocyte and then go to the bloodstream. Obviously, there are exceptions to that, but it is sterile. Okay, moving on. There's nothing much that you guys have to know over here. Let's move. This is a picture that your professor used in her slides, and this is basically a comparison of the surface area. So as you can see over here, the enterocyte is 600 times more, like in terms of surface area, it's larger than a regular cylinder, or if you see the intestine as a regular cylinder. This is just concepts, you don't have to memorize anything over here. But this is a huge advantage while absorption is occurring. As you can see, the intestine starts as a cylinder, then there are folds, which increase the surface area, then there are villi, which further increase the surface area, and then those villi have microvilli. Again, increasing surface area, pretty self-explanatory over here. Now, absorption. Angelica went into two types of transport while, absorption is happening. So there is microtransport, which entails active transport and diffusion. Active transport can be primary and secondary. So primary mainly involves ATP. Secondary involves the difference in a concentration gradient or like an electrochemical gradient. So usually the ion that we will be seeing over here in secondary active transport would be sodium. I need you to keep secondary active transport in mind, and I will be asking a question on this later. Now, diffusion can be passive. This shouldn't be active, sorry. It should be passive or facilitated. So facilitated is through a transport molecule, and passive is just passively through the membranes. You don't need a molecule to go through. Now, macro transport is exocytosis, penocytosis, and endocytosis. You guys probably know about these, but I will be talking specifically about endocytosis. It does not normally occur in adults, but it is involved in the absorption of B12, specifically in the ileum. You have to remember this. B12 is only absorbed in the stomach and the ileum, and I remember there being a question in our final about this. Um, that they resected like a part of the small intestine, like like distal intestine. They didn't specifically mention the word ileum. So that part was removed, and that caused the deficiency of a vitamin, and they asked us like what vitamin was affected, so that would be B12. B12 is again only absorbed in the stomach and ileum. You have to remember this. And apart from that, endocytosis also occurs in babies, specifically breastfed babies, because they don't have their own immunity, and immunoglobulins are absorbed using this process. Moving on, there are different sites of absorption. So there is the stomach, there is the small intestine, and there is the colon. I just want you guys to remember what each absorbs. And Angelica did not go into specific detail into any of these. She mentioned that the stomach is facilitated for its job in absorbing ethanol, alcohol, NSAIDs, and aspirin, like medications, because it needs to protect itself from these harmful substances. And the duodenum, jejunum, and ileum, they are the small intestine. Again, this is where 95% of water is absorbed. So even though it says the colon absorbs water, but the main absorption of water happens in the small intestine. I want you guys to remember that. That was also a question, if I'm not wrong. So where is water mainly absorbed? It's absorbed in the small intestine. Don't mistake it for the colon. Okay, moving on, we will be talking about carbohydrates, how they are digested, and how they are absorbed specifically. So carbohydrates, they can either start off as starches, lactose, or sucrose from the food that we eat. Let's get into starches first. So starch, the digestion of starch starts in the mouth itself, but that is not where the main digestion takes place. The main digestion takes place in the GI, but it starts in the mouth using ptyalin, which is salivary amylase. So 20 to 40% of the starches are digested in the mouth itself. Then the pancreatic amylase does most of the job in the GI. Okay, starch is broken down into maltose. Okay, it entails 3 to 9 glucose polymers. Now, maltose can further be broken down into glucose. It forms 2 glucose molecules by using the enzyme maltase. Now, maltase, lactase, and sucrase are brush border enzymes. So as you can see, the diagram shows that like in a bracket, it says intestine. Oopsies, sorry. Um, I cannot remove that. Sorry. Okay. Uh, it says intestine. So basically, all of these are brush border enzymes. So what this means is that maltose, lactose, and sucrose is mainly being digested and absorbed in the small intestine itself. So digestion also takes place in the small intestine of these three substances. So maltose breaks down and forms 2 glucose molecules. Lactose breaks down using the help of lactase and forms glucose and galactose. Sucrose breaks down and forms a molecule of fructose and glucose. You need to remember the products, okay? Moving on. I can't move on. Sorry. Now that they are digested, the 3 molecules, fructose, galactose, and glucose, need to find a way into the enterocyte and then into the circulation through blood. Okay. There are different transporters for these, and this is really high yield and important. I will be starting off with fructose. Fructose in the name, it says F, so F, and 5 has an F. So that's how I like to remember it. So GLUT5 will be the transporter for fructose. So now, can anyone tell me what type of transport this is? Facilitated diffusion. Yes. I'm sorry, Saif, I didn't read your question early on. Is starch also broken down into lactose to ptyalin, or is it only specific to maltose? I am not too sure about that, but I think it's specific to maltose. And stick to the information that was given to you in the slides. Yeah, Angelica did not mention it, so it's extra. You don't really have to know. Okay. Um, so this is facilitated diffusion. Great. Facilitated is because it's using a transporter molecule. Now, then it goes into the lumen of the enterocyte, then it can be transported into the circulation using GLUT2 transporters. GLUT2 transporters are used for all three, so there's a competition between all three to use those. I think it's really easy to learn, and it's it's really high yield. It's important. Um, Now, moving on to glucose and galactose, they are done a different way. They are absorbed a different way through a symporter. So Angelica did not really use these terms, but our professor did. We had AJR back in the day. So he used the term symport. Now, symport is a type of secondary active transport. And now, what did I tell you guys about secondary active transport? It requires a concentration gradient. Now, the concentration gradient over here is of the sodium ion. And how is this concentration gradient maintained? It's maintained through the sodium potassium pump over here. Um, I'm sorry. This is really important. Again, high yield. The concentration gradient is maintained by the sodium potassium pump. Um, So what the sodium potassium pump does is basically whatever sodium enters the enterocyte, it goes into the blood, leaving a deficit of sodium in the enterocyte itself. So there is no option but for sodium to move from the lumen of the GI into the epithelial cell or the enterocyte. And while doing so, it drags along another molecule. This molecule could either be glucose or galactose, and as we will see later on, it can also be an amino acid. And it's really important to know the name again, SGLT1. Both glucose and galactose are competing for this transporter. Okay? And then from then on, GLUT2 transports all three: fructose, galactose, and glucose. Do we move? Is everything clear? Okay, thank you so much. Now, that is the end of carbohydrate digestion and absorption. We will be talking about a pathology, which is lactose intolerance. So does anyone have lactose intolerance over here? Because I personally do, but I try to hide it under the rug because honestly, it's embarrassing, right? Um, why am I beefing with milk? Realistically, okay? Now, lactose intolerance is due to a lack or non-functional lactase. So it can either be absent or non-functional or not normally functioning, okay? Lactase, again, is a brush border enzyme, as I mentioned before. So what that means is that it's mainly situated near the small intestine, right? Near the microvilli, near the villi. Um, so what happens is because lactase is non-functional or it's absent, or it's just not working as efficiently as it should be, lactose accumulates in the intestine. And because of this accumulation, the osmolality of the intestine increases, so there is water retention. Do you guys get that concept? We studied it in foundation as well, osmolality. So because the concentration of the substrate is high, substrate being lactose, it drags along water with it, which causes bloating and eventually watery diarrhea. That's why you sit in the bathroom for hours after you eat dairy. However, it is important to know that there is no damage to the brush border. So if you take a histological slide, you take biopsy of the small intestine and you go under the microscope and see it, there is no actual damage to the brush border. This is a purely enzymatic abnormality. It's nothing regarding the intestine itself. Now, how do we diagnose this? We can diagnose this by using the H2 lactase breath test. However, it might give you false positives. This is just like it doesn't confirm that you have lactose intolerance. It just pinpoints you to a certain direction where you can like do further tests and figure it out. So why H2 specifically? Now, you guys know that there is normal microbiota living in your gut. So there is bacterium, which is normal, and it lives in your gut, and it is helpful more than harmful, but it can become harmful if it feeds on your food, obviously. So because there is an accumulation of lactose in the gut, the bacteria feeds on on the lactose, and it releases hydrogen as a result. And this hydrogen can be measured in your breath, and it can lead you to a diagnosis, basically. So the treatment for this is, all in all, avoiding dairy, or you can use like supplements, lactase supplements before you eat dairy. You know, you're gonna eat dairy, so you pop a pill, and it can help. Moving on, we will be talking about proteins. Now, proteins have three main places of digestion. So I told you carbohydrates, the digestion starts in the mouth itself. Proteins, it actually starts in the GI lumen. So it is exposed to stomach pepsin and pancreatic proteases. Pepsin specifically digests or breaks down collagen. We don't have to get into too much detail. Then another place of digestion would be the brush border itself, which deals with oligo and dipeptidases. So oligo and dipeptidases, what does that entail? Oligo means more than three, and di means two. So peptidases, which means that these peptidases will be dealing with proteins that. Thank you so. Sorry, guys. Thank you for being patient with me. Again, back to oligo means more than three. So these peptidases will either be dealing with peptides, proteins that have more than three amino acids in their chain, or two amino acids in their chain. And then the cytoplasm of mucosal cells, they deal with di and tripeptides. So proteins with two amino acids or three amino acids in their chains. Now, peptidases can further be classified into exo and endopeptidases. So what this means, you can sort of get an idea with the name itself. So endo means inside the chain. This is how I like to remember it. So as you can see over here, this is a long peptide. What endopeptides will be doing is that they will be breaking down the bonds between um the amino acids themselves, like not in the middle, like not at the terminal ends, if that makes sense, right? The terminal ends would be the amide end and the carboxyl end. Now, exopeptidases, they focus more towards the outside of the peptide. So there are two exopeptidases, the aminopeptidase and the carboxypeptidase. The carboxy deals with the carboxyl end, and the aminopeptidase, again, deals with the amino end. I think that's really self-explanatory, but please do know these details. They're very small details. Any questions at all? No? Okay, we will be moving on. Thank you. Mm, I'm sorry for being so haphazard. Anyways, So there are different transporters for basic, acidic, and neutral amino acids. We don't have to go into detail. Just know there are different transporters. She didn't specify any names. The only names that she specified were for the carbohydrate transporters, which is SGL, T1, GLUT5, and GLUT2 that you have to remember. So there are different ones for amino acids. We didn't go into details. Again, this is symport, which is secondary active transport. And the amino acids are co-transported with sodium ions. However, di and tripeptides, they are co-transported with hydrogen ions. Okay, so know this difference. And some smaller peptides that haven't been broken down, or you can call them oligopeptides because they're more than 2 or 3 amino acids in a chain. They just undergo transcytosis, which is basically just moving from one lumen, the GI lumen, into the next, into the blood. This is transcytosis, or you can say a combination of endo plus exocytosis. Okay. So yeah, just know that. Now we will be dealing with the complications of, more protein related pathologies. So one of these is acute pancreatitis. So titus is inflammation. So this would be inflammation of the pancreas themselves. Sorry. And why this occurs is basically the enzymes, they activate prematurely. They activate in the pancreas instead of the GI itself or inside the intestine. And this eats away at the pancreas. Okay. So the premature activation of enzymes inside the pancreas instead of the lumen of the GI, one of these enzymes is trypsin. This disease can be associated with alcoholism, biliary tract disease, and in children, it can be associated with cystic fibrosis. It can also be caused because of overeating. So because you're overeating, you're exposed to a lot of enzymes, you're releasing a lot of enzymes, and this can damage the pancreas. And the treatment usually is fasting. This is really important, or antitrypsin or trypsin inhibitors. I don't know if you need to go into the specifics or the statistics, but um, in women with biliary tract disease, the statistics show that women are more susceptible to acute pancreatitis, and in people with alcoholism, men are more susceptible. So the ratio is 6 to 1. 6 men are affected, whereas 1 woman is if they're alcoholics. And the ratio for biliary tract disease is 1 to 3, 1 being men, 3 being women. So 3 women versus 1 man with biliary tract disease would be, affected by acute pancreatitis. I'm pretty sure it's not that important, but please do remember just for your own sake. Now, moving on to cystic fibrosis. Cystic fibrosis is a genetic defect in the CFTR transporter. This is a transporter dealing with chloride ions. So what happens is that basically, usually, the delta F508 gene is the most commonly associated gene with this genetic defect, but it's not always this one. This is the most common one. I would like you guys to know this. It comes up a lot later, but Angelica did not mention this. Your professor did not. So what happens is that because chloride ions, they are prevented from being transported back into the sweat duct. So this is the sweat duct, sorry, sweat gland. They cannot be regulated, and chloride stays inside the sweat duct, and it causes sodium to stay with it because chloride is a negative ion. It attracts positives, and it attracts the sodium ion. So NaCl is salt. It causes the sweat to be very salty. So what happens is that chloride is not regulated to go back inside the body. It causes the retention of sodium as well. And the only way to regulate it is by sweating. So that's why the sweat released is really salty. Does that make sense? Any questions? I will. Okay. Okay. I'll be moving on. Thank you guys. Now we will be moving on to lipids. This is the main mechanism. This picture speaks for itself. If you know this, I think you should be good. But the main processes in lipid metabolism and absorption is first off emulsification, then enzymatic digestion, then reconstruction of those lipids, and chylomicron formation. So what is emulsification? First off, you start off with a huge lipid droplet. Then bile salts are added to the picture. Bile salts are amphoteric molecules that are both hydrophilic and hydrophobic. They are hydrophobic at one end, hydrophilic at the other. It's sort of like, your cell membranes, basically. So what they do is that they form a membrane and they form a structure called a micelle, and they entrap lipids into a micelle, breaking them down. So basically, they're not broken down chemically, but they're formed into smaller droplets. So big droplets form smaller droplets, which are called micelles. Now that they're smaller, they can be digested more easily. So now lipases, different lipases are added to the picture. We'll get into what lipase later. And then, so now that they're broken down and they're refined, so I would like to call them refined micelles, those micelles can go into the enterocyte. Some of the fat is directly changed into short chain fatty acids, which can go directly into the enterocyte, and it doesn't need to go through a micelle. But we'll talk about that. And then inside the enterocyte, there is further reconstruction of the lipids, and we basically sort out what lipids that we want and in what formation we want them, and in like how long the fatty acid chains should be and stuff. That is done inside the cell. And a chylomicron is formed, and then the chylomicron can either go into the blood circulation in the capillary, or it can go into the lacteal, which leads to the lymphatic circulation. Moving on, we'll be talking about lipases. So I just copy pasted this off of your slides. It is really important. There are three different types of lipids that you will be dealing with from your food itself, and that need to be broken down into simpler forms. So triglycerides, cholesterol esters, and phospholipids. Triglycerides are broken down by lingual, gastric, and pancreatic lipases. They are broken down into monoglycerides and fatty acid chains. So now these fatty acid chains can be short fatty acid chains or longer ones, okay? We'll get into that in a bit. Monoglyceride, it can be glycerol, it can be other products. Now, the cholesterol ester is broken down by the cholesterol ester hydrolase. It's broken down into one cholesterol molecule and one fatty acid. And phospholipids are broken down into lysolecithin and fatty acids. So the final products of lipid digestion are cholesterol, monoglycerides, fatty acids with ranging fatty acid chains, like the chains itself, and lysolecithin. It is really important to know the byproducts. Please know them. Moving on, this is just directly from your slides because it's a really important concept that I didn't want to mix up. So what happens is that what we're talking about over here is we're talking about how short chain fatty acids are absorbed versus long chain fatty acids and monoglycerides. So the lipases, they do their job. They break the triglycerides and the cholesterol esters and the phospholipids down into cholesterol, isolectin, fatty acids, and monoglycerides. Now, the fatty acids, they range in their chains, so they can be long or short, as I mentioned earlier. The short ones, they can just go into the enterocyte by simple diffusion. So they can dissolve into the intestinal chyme and they are reabsorbed by simple diffusion, as you guys saw over here. Okay, over here. This is just by simple diffusion. It doesn't have to be in the form of a micelle. Longer ones, they have to be in the form of a micelle. The micelle includes other products like monoglycerides, lysolectin, and other lipid-soluble vitamins, which are vitamins A, D, E, and K. Vitamin A, DEC. Just really remember this. This will be a concept that is repeated later on. The lipid-soluble vitamins are A, D, E, and K, and they are part of that micelle. So along with biosalts, they are transported into the brush border where they diffuse out of the micelles into the absorptive cells, okay? So the mechanism, basically what I'm saying over here, the mechanism of absorption for short-chain fatty acids and long-chain fatty acids is different. Now we move on. We said that there are three main processes to this whole thing. So first there's emulsification, enzymatic digestion, then there's reconstruction. So now we will be talking about reconstruction. So what happens over here is that now the micelle contents are in the lumen of the enterocyte. We need to sort out what fatty acids we want and how long we want the chains. This all happens over here. And we also add the lipid-soluble vitamins, vitamin A, DEC, and apoproteins as well. Okay, and now we form a chylomicron. So I would say a chylomicron is basically a refined, reformed version of a micelle that is ready to be put out into the body, and it has the most efficient lipid byproducts. Okay, now exocytosis of the chylomicron happens out of the absorptive cells into either lacteals and or into the blood circulation. Now, if it goes into the blood circulation, it mainly circulates as LPLs or HCLs, LDLs and HDLs. We'll get into that. So first off, I want you guys to know about this enzyme, LPL, lipoprotein lipase. This is an enzyme present in the epithelium cells of the adipose and the liver tissues. So what it does is that it breaks down chylomicrons, and it basically makes the lipids available in a form that can be absorbed by the cells for use. So it breaks it down into triglycerides or fatty acids or whatever form that the cell can actually absorb, because the chylomicron itself is inefficient to absorb, if that makes sense. So we want lipids either in the lymphatic system or free, which is what the lipoprotein lipase does, so they can be taken inside the cell. When the chylomicron is circulating, it's either circulating as LDLs or HDLs, which is low density lipoproteins, or HDLs, which is high density lipoproteins. You'll get into that later. We studied about this more this year. But what I want you to know is that the liver makes LDLs and HDL is transported to the liver. Does that make sense? So the liver makes the LDLs, which takes triglycerides to the cells and makes it available in a form that can be used by the cells, and HDL is the one going to the liver. So LDL leaves the liver, HDL goes to the liver. This is an important concept. Okay, moving on. We will be talking about, we have only two last concepts left. We will be talking about malabsorption and celiac disease. So malabsorption can, usually we talk in the terms of fat, because fat is measurable in the stool and, like, you know, the byproducts of whatever, and it can be measured. Malabsorption can be caused by three different disorders. It can be motility disorders, digestion disorders, or absorption disorders. These sound very similar, but they are pretty different. Motility is an issue in movement. So it can be that you're emptying out your bowels too fast or too slow. So it's taking a lot of time in digestion or very fast digestion. Or no, not digestion, motility, movement. So it doesn't allow for proper digestion. So if you're going too fast, that is a problem because you're not able to absorb all the nutrients that you can from the food that you have eaten. If you're going too slow, then the absorption is going too slow and the normal microbiota can feed upon the food that you have eaten and cause an issue regarding that. And these motility disorders, as Angelica mentioned, they're mainly neurological. Now, digestion disorders are disorders by the enzymes, so mainly lipase is affected. So it can be caused by pancreatitis or cystic fibrosis, right? So these are enzymatic disorders. There is no problem in the intestines themselves or the GI themselves, okay? And then there can be absorption disorder. This is a disorder in the intestine itself. So either the brush border is damaged or the intestine is damaged or you have removed part of the intestine, okay? So for example, if you remove the ileum, right, you won't be able to absorb maximum B12. That would be an absorption disorder, okay? Moving on, we will be talking about a specific type of absorption disorder, which is SPRU or celiac disease. Now, celiac disease, this is caused by genetic disposition. You don't need to know all of this. Just know that there are certain immunoglobulins and genetic factors that cause the brush border to be very small. Now, because it's really small, this is a normal brush border, this is very tiny. Because of this, obviously, the surface area is decreased, which prevents the maximum absorption of the substances. And it causes bloating, it causes digestion issues, it causes pain, and it is often overlooked. And what Angelica mentioned is that it is the commonest malabsorption syndrome in the US. And people usually don't find out that they have celiac disease until way later on. People with celiac disease are given advice to be on a gluten-free diet. Why is that so? That is because that gluten is really hard to absorb for them, and it won't be able to absorb in time, basically. So to prevent the irritation that comes with eating gluten, they are put on strict diets that are without like gluten bread and stuff like that. Apart from that, people can also face anemia due to this because this is an absorption disorder. You can't absorb iron and other nutrients, which causes further complications. So basically what I need you guys to know over here is that it decreases the area available for reabsorption, and there is a decreased production in intestinal digestive enzymes because the brush border itself, it's made up of different cells which release these digestive enzymes, and if they're not present, obviously there will be a lack of enzymes. And that is all. Thank you guys so much for your time. If you have any questions, I'll take it right now, and please feel free to leave your um comments in this QR code.